All right, so it's my pleasure today to introduce Dr. Herrick Brown, who's going to present this topic today. He serves as the curator of the A.C. Moore Herbarium, Department of Biological Sciences here at the University of South Carolina. Prior to his current appointment, he served as assistant botanist for the South Carolina Department of Natural Resources Heritage Trust Program, and acted as an official liaison between the herbarium and the SCDNR. In addition to his involvement with the South Carolina Plant Conservation Alliance, Dr. Brown serves as a member at large for the South Carolina Exotic Pest Plant Council. He's a faculty affiliate of the Mark Catesby Center and is actively engaged in related projects within the Plants and Planters and Historic Southern Naturalists. And that is aimed at digitization of biodiversity collections and related archival materials in the state. Regionally, he serves as the data manager for the Southeast Regional Network of Expertise and Collections portal. Dr. Brown's research goals involve mobilizing biodiversity collections information for use in salient research applications. For example, the herbarium specimens provide a snapshot of what plants are doing, whether they're flowering, fruiting, just growing a new leaf at a particular place in time. This source of information, when you consider it across multiple specimens, can be used to understand patterns and species distributions of seasonal timing of reproductive events. Since some of the herbarium specimens were collected many years ago, as far back as the 19th century, it is possible to then document changes in the time of seasonal expression or phenology in plants, which has been attributed to changes in climate conditions. So specifically, Dr. Brown's research interest involved dispersal potential for plant species endemic to the Southeastern United States, species distribution modeling, and the potential impacts that forecast climate conditions and what they might have on conservation efforts aimed at habitat preservation and plant community composition. So it's my pleasure to turn it over and welcome Dr. Brown for his presentation.
Cool. Thank you, Dr. Olatosi, and thank you all for joining us today. I'm excited to present to you tales from the Botanical Crypt and how our herbarium collections, when digitized, can help with new sorts of research applications that traditionally were not possible. So without further ado, I'd like to eschew any obfuscation over what a herbarium is by telling you what a herbarium is not. It is not an apothecary or an herbal repository for ground up plant parts. It's not an arboretum, which would have living specimens of trees. It is not a botanical garden, which again is more of a formal planted landscape, a conservatory, or a greenhouse where you would have sort of hothouse plants and that sort of thing. In fact, a herbarium is a collection of preserved plants. Sometimes you might have the entire plant pressed and glued to a sheet, or if the parts of the plant are too bulky or too large to fit in our standard 11 by 17 inch sheet of paper, we might just house them separately in little bags with a corresponding label that associates them with the pressed specimen. Most of the time these things are dried. Sometimes we have wet collections, depending on what resources are available to the individual herbarium. And it's essentially like a plant library. It's a source of primary reference materials. Each specimen has a label affixed to it, and that records all the collecting information associated with when it was captured from the wild, so to speak. So in a sense, it's more like a plant morgue than anything else. They have toe tags that give us who collected it, so the murderer, and they have date of collection, which would be sort of like the time of death, very, very analogous to a plant morgue. The process of making specimens, excuse me, is very manual. It involves field trips, which is a lot of outdoor work. It's very enjoyable for some folks. And the botanist usually carries with them a sort of field press, which might be a couple of loose sheets of cardboard between which they have several loose leaves of newspaper. And as specimens are collected, they will be stuffed in between those sheets of newspaper and cinched down, possibly with something like a parachute strap to kind of keep them flat. Once we get back to the herbarium, the pictured at left is a formal press where we use packing materials such as, it's almost like the underlayment for carpeting, and corrugated cardboard sheets along with blotter paper. Blotter paper is basically just a thick cotton rag paper. The foam sheets allow for absorbing some of the topography of the different specimens. Woody specimens tend to be, there's more of a stick-like hard material to it, so we want to kind of account for any sort of bumps that they might otherwise inflict on their neighboring specimens in the press. The corrugated cardboard, because of the channels that run through it, allow for free airflow between the specimens, and the blotter paper is just an absorbent material that helps wick away the moisture from the plant. And all these steps are very critical in terms of preserving the specimen because, especially in the Southeastern United States where we have a lot of high humidity, they can quickly begin to mold once they're collected. The other thing that we do once they're in this press is that we'll stick them in a drying oven. Some herbaria, say in the Great Plains states where humidity is really low, don't really need to use an oven. They simply set the specimens out in a similar kind of press, but put large box fans in front of them so that there's a constant airflow. And what we found is the best way to preserve the specimens is kind of like a good barbecue, long and low. So we put them in an oven that's roughly about 100 degrees Fahrenheit and leave them in there for 3 days to maybe a full week. And this process is sufficient to kill any potential plant pests or bugs that would otherwise eat and destroy the specimen. And it's also adequate for preserving some of the DNA, so we can do some sampling of the specimens once they're finished being processed. After they come out of the oven, there's another manual step where the specimen has to be carefully arranged on the finite amount of space that we have on the standard herbarium sheet, which is again roughly 11 by 17 inches. They're glued to the sheet. We use Elmer's. There's some debate about which glues are best to use, but basically it's a polyvinyl acetate based glue that's relatively inert and has a pH somewhere around 7. So it's neutral. Again, it involves sort of a careful arrangement of the plant specimen so that it all fits on the sheet, and we sometimes will flip things around so that you can see both the upper surface and under surface of leaves. Sometimes we will do sort of a dissection so that you can see the inner parts of a flower or fruit. Any of those sort of illustrative type characters that would help in diagnosing what the species is, we try to reveal those during this process. And of course, the typed up specimen label will also be affixed, usually in the bottom right corner of that sheet. And once it's been mounted on the sheet, it goes into a digitization queue. So we use a database that has fields that are standardized and internationally recognized as a standard for how the different data bits are parsed out. And that still is also sort of a manual re-entry of the data process. There are some workflows now that have sort of streamlined this so that there's one-stop data entry where the specimen label is generated by the collector. It can be printed out and then also automatically uploaded to the database. So we're getting there in terms of making things move faster through the queue. Once the specimen has been fully entered in the database, we then go to an imaging queue. We use these fancy light boxes that give us almost zero shadows on the specimens, and a 14 to 18 megapixel digital camera. The end result essentially is a specimen that we can zoom in and get the equivalent of about 40 times magnification, which is the same sort of magnification that we would use in a dissecting scope when we're looking at specimens, looking at the physical specimen in the herbarium. And the data are parsed into individual fields. And this kind of, it's a data management trick that helps us make the specimens more useful, because the way they are housed in the herbarium physically, we can only sort of arrange them in so many ways before they're no longer useful, and we exhaust the possibilities for other ways of arranging them. In our collection across the hall from where I am now, we have it arranged alpha order by family name, and then within the families they're alpha order by genus, and then within the genus alpha order by species. And they lay flat in similarly sized folders, and we have a bit of a geographic coding scheme going on where plain colored folders represent material collected from South Carolina and naturally occurring material from South Carolina. We have green folders that are used for anything that's cultivated in South Carolina. And then we have orange and blue folders, which are from material collected in other Southeastern states or anywhere else in the world. So our collection is quite broad in terms of its geographic scope. We've got specimens from Vietnam, the Middle East, Europe, Africa, Central America. But by far our focus being that Columbia, South Carolina is our home. We have the greatest representation of our collection is South Carolina material. On the slide that you're looking at now, the little labels pointing to the label elements are using a Darwin Core standard name for those fields. And you'll notice on the right-hand side, bottom right-hand corner, we have recorded by. That's basically the person who collected the specimen, and the event date would be when they collected it. The barcode value is critical in terms of managing our association between the digital image and the metadata, as the file name matches that barcode value. Let's see. So the end result, once we've got all that processed, the specimen's been glued to the sheet, it's been imaged, and all the metadata have been entered, you can then go to an online portal and search through all those records, and they'll appear as they do on the left-hand side of your screen in sort of a list view. And if you click on the full record details, you can see all the metadata and the associated specimen images. So traditionally, before biodiversity collections in general had initiated their efforts to digitize their collections, the way these sorts of data would be accessed was very painstaking and very slow. The first step of even identifying where you might find specimens of interest largely involved either direct phone calls or letters being mailed to inquire whether or not any relevant material was at a particular institution. Literature searches were sort of a trick that could be used where you could scan any sort of published accounts and look for herbarium acronyms. So I've highlighted USCH, which are the internationally recognized call letters for the A.C. Moore Herbarium here at U of SC. And that acronym is maintained by a registry service called Index Herbariorum. And anyway, so you could scan the literature and look for mentionings of herbaria that might potentially have material that you want, and then initiate a request for a loan. Those loans, one way or the other, even today, are sent out through USPS library rate. They get boxed up very carefully, tightly packed, so there's no shifting of the specimens in transit, and as hermetically sealed as possible. So we use a lot of packing tape to seal up all the cracks on the shipping package. Traditional uses for herbarium specimens were relatively limited, mostly for comparative analysis, sorting out which one of these things is not like the other so that we could organize the collection in a taxonomic sense. And then other sorts of studies aimed at potentially describing new species would come out of that. A few other sorts of things that I don't have mentioned on the slide here would be conservation efforts looking at occurrences of rare species, and conservation agencies like South Carolina Department of Natural Resources would use the herbarium to find previously undocumented populations for these rare species, and then attempt to do site revisits based on the label information. And so they actually, that agency, DNR, actually maintains their separate database, which is more based on field observations, but they try to incorporate any associated herbarium vouchers along with whatever records they have as well. And so some of the challenges associated prior to our digitization efforts were chiefly the fact that access to these primary reference materials was extremely difficult. We refer to it affectionately as dark data. The herbarium cabinets that we use have they're steel cases that stand probably about 6.5 feet tall and have approximately 26 individual cubbies or shelves in there, and they might hold about 1,400 specimens apiece, depending on how bulky the individual specimens are. And then that is all sealed with a nice steel door and a neoprene gasket. And the idea there is that we want to keep any fluctuations in humidity away from the specimens. We want to keep any potential insect pests like cigarette beetles out of there. And so they're closed up, they're behind closed doors. And if you were to come over and have a tour of the herbarium, you would just see a maze of cabinets. It wouldn't be very exciting. So access to the collections has been difficult, and we know that there is a treasure trove of data within them. So the way that we are able to make those data available is through digitization. And there's an added benefit to that, which is that preserving these specimens as indefinitely as we can is a little bit easier, because any potential user can view the digital information without having to access the physical specimen and potentially running into some sort of situation where a leaf might be snapped off accidentally or coffee might be spilled on it, those sorts of things. And because of the sort of limited access to these sorts of data, the traditional taxonomic studies like descriptions of new species quite frequently would take up several decades. The holotype that's pictured at left was originally originally observed and collected in 1978, but at that point in time, the taxonomists only knew that it was something different. They didn't know what it was. It could have been a disjunct species from points further west. It could have been a hybrid of some sort. So there was a lot of uncertainty, and nailing down the fact that it was indeed a new species took a lot of loans from other herbaria to do comparative analyses, and a lot of careful scrutiny at the character states that this particular species possesses in order to determine how it is actually different from all the other species in that genus. So now the herbarium data can be accessed in a variety of ways digitally. On the top left are what we call data aggregators. GBIF is a global facility that aggregates data from all over the world, and they incorporate a variety of biodiversity collections as well. So they're not limited to just herbarium specimens. They would include lepidopteran collections, mammalian collections, those sorts of things. iDigBio is the national resource for biodiversity collections, and they are sort of a partner of the project that I'm involved with, and they help aggregate data from all the regional consortia across the United States. The other sort of banners that you see over there on the left are representative of some of the regional herbarium consortia that are available online now. And while they all have their own different brands, if you were to visit any one of those individual websites, you are effectively searching the exact same amalgamated database. So CERNEC is the Southeast Regional Consortium of Herbaria, basically. And if you've visited the CERNEC website, by default, you can actually search the Southwestern Network, the Texas-Oklahoma Network, and all the others as well. And the same would hold true if you visited the Intermountain Regional Herbarium Network. You would just have that sort of Intermountain region brand, but you would still be able to search all the other regional consortia. So anyway, online portals are a way of accessing them. Depending on individual herbarium's level of IT support, they may have their own website, and you might be able to access more detailed information from it. And of course, email is sort of it's more commonplace now, but it was definitely a useful tool when prior to having digital specimen information online. So I'd like to focus a bit on the CERNEC project, which again, I served as the data manager for this portal. And it began as a research coordination network that was funded through NSF. We had several annual meetings that were held in conjunction with the Association of Southeastern Biologists. And the initial thoughts were, and this is kind of reflective of the organization's name, were that we would be sort of an all taxa group, not just limited to herbaria. And the sort of the default was that most of the people that attended these meetings ended up being herbarium curators. So the rest of the taxa just didn't have enough representation for us to really focus on what their needs were. And it may have been that, you know, herbaria had some advantages in that our specimens are very flat and not pickles. So it's a little bit easier to kind of digitize things like that. Eventually, we planned through those research coordination network meetings to submit some proposals to a relatively new program at the time called Advancing Digitization of Biodiversity Collections. And the initial round of proposals actually is what funded the iDigBio national resource. And then there were a few what they call thematic collections networks that were funded at that initial phase. We attempted to get funding to form the CERNEC portal as a thematic collections network at least 3 times, and finally we got it right. And it was a beast because we had collaborators from 13 different states across the Southeast involving 93 herbarium collections, and forgive me a second. 93 collections across the Southeastern United States, and the way we had to submit that proposal ultimately caused Fastlane to crash. So we ended up having to submit everything manually the day after because there was no other way to get all the information to NSF. On the 3rd time, we were funded, and NSF said, "Hey, great news, you're funded." And then they said, "But we need you to reduce your budget by nearly 40%. So we were still held to the same numbers that we had promised to deliver with almost half the budget we had asked for. So that was kind of a challenge. And the title of this project was The Key to the Cabinets, Building and Sustaining a Research Database for a Global Biodiversity Hotspot. That would be focused on the Southeastern United States. And in particular, our focus were endemic plant taxa that were endemic to the Southeastern US. So that kind of helped us limit our numbers that we were, you know, on the hook to deliver as part of this now funded project. The goals were to image about 4.7 million specimens, and that was sort of the design, was to go at it from just capturing images up front and not worrying about developing the metadata because we had a very novel approach to involving citizen science, our outreach portals, something called Notes from Nature, wherein the herbarium specimens will be served up to a virtual citizen scientist who then would look at the specimen label and transcribe that label information into the various data fields that we wanted them to populate for us. And that was a very effective method, at least in some states, but depending on how their prior digitization efforts had either existed or not. Arkansas, for example, was able to enroll native plant societies, and their membership was, obviously very gung-ho about helping make these data available and developing the metadata for the associated images. And so it was really rewarding initially to see this project grow from just a handful of sort of pilot collections that we included in 2013 to several dozen a few years later, and then have collections on board something like 60,000 records from nothing prior to that. The project also involves a collaborative georeferencing effort. So we have a sort of a liaising software package that talks to Geolocate, which is a biogeomancer. It has essentially a database of geographic names, and it assists the user in reading a verbatim locality and trying to approximate where the coordinates might be, and then the human user evaluates the results returned from Geolocate to either move the locality slightly or assign some sort of uncertainty radius around it. So this is kind of just a real quick graphic showing how the project progressed. Our onboarding of collections ramped up significantly during the middle of the project. The project actually ran from 2014 to 2018. It was supposed to be a 4-year project. I mentioned that we had a few pilot collections that we put into the portal in 2013. That was sort of more of a proof of concept effort there. And then we got a no-cost extension that took us into the middle of 2019. And so you can kind of get a sense of some of the collections. As I recall, in Tennessee, they took their time sort of making sure that they had their game plan and all their ducks in a row. And by 2016, they just added a ton of new specimen records. And so that's why you see that sort of big hump in the middle there. So we've exceeded our project goals. We now have about 118 collections. Our estimates are within the Southeastern United States that there are at least 233 herbaria. So we have a little ways to go in terms of including all of them. We have over 5 million specimen records. So we've punched through that 4.7 million initial goal. And our citizen science components and georeferencing are still ongoing. And you can sort of see that in our summary statistics there, with the occurrence records being a little over 5 million. And we only have about 10% of those with latitude and longitude assigned to it. But a great deal more have images associated with them. So the strengths of this project involve the diversity of our personnel and the collections. They all had subtle nuances and novel ideas and different approaches to how we would accomplish this. And while we set out to establish sort of a standardized workflow, invariably there was always some exception to that rule. But our communication was so well refined at that point in time because we had several years of that research coordination network that preceded this effort, that we were very capable in terms of sharing how we arrived at solutions to the various problems. And that played into sort of, our flexibility in terms of how we approach the prescribed workflows that we had published. And then also we sort of took I don't know, I guess it's sort of democratic approach to it. There was, a lead PI who was out of Appalachian State University, but each individual state had a state lead PI who would coordinate with the herbaria in their individual states. And that worked out really well because I don't know, the way the herbarium community works apparently is that we all are just more familiar with our neighbors within our geographic bounds. So it was quite an experience. Some of the challenges that we had though were cat herding. So we always had somebody who wanted to do something different for no apparent reason, or despite all your best efforts to recommend best practices, would deviate from that advice. We had a few cases where folks were taking jobs at new institutions midway through the project, so we would have to coordinate with them to train incoming personnel, and ensuring sustainability, which is part of the project title. We're in a pretty good position at this point in terms of data storage. We've got assurances from Arizona State University, where the actual database is maintained at this point. They have funding through, I think it's another NSF-funded project that's related to NEON, which is the National Environmental Observation Network. And that funding, I think, is for at least the next 20 years or so. And then all of our images are served through something called Cyverse, which was previously called iPlant, and they afford us almost triplicate backup of all the specimen images, and those are on servers that are at the Texas Advanced Computing Center and Arizona State and maybe one other location. So it's also geographically distributed. So what's next? Well, we decided to hold a stakeholders meeting, and that's the lead PI on the left. No, I'm just kidding. It's we tried to evaluate what we needed to do in terms of making the portal more relevant. And obviously, since we're lagging behind in our georeferencing, we need to step that up a bit. We only have 10% of the occurrence records with latitude and longitude, and that kind of handicaps the ability for us to apply these data in other novel ways, especially when it comes to like species distribution modeling and those sorts of things. We're doing okay as far as the specimen images are concerned. It's a little bit behind, but not too far behind there. And then this other question of the 115 remaining herbaria in the region, which might include another 5 million specimens. And a lot of those tend to be smaller collections. And so we ran into this question of what's the relevance of these smaller collections? Do they really have anything unique to offer that you wouldn't find at some of the largest herbaria in the world, like New York Botanical Garden or Paris Natural History Museum, those sorts of things? So I did like a little quick case study on some of the smaller herbaria that are within the Cernak region, and found things like this specimen of dwarf flowered heartleaf, which is another common name for this would be wild ginger, even though it's not technically a ginger, and bunched arrowhead, Sagittaria fasciculata. And USC Upstate in Spartanburg has these fantastic specimens that with a really cool specimen label that shows exactly where they were collected in Greenville County. And they ended up being on some land that was right adjacent to a heritage preserve, which was, the preserve was actually there because of these plants were known to be on that property. But these specimens were collected from the adjacent property that was slated for development. And so there was this big uproar involving Upstate Forever and the Heritage Trust Program and U.S. Fish and Wildlife, because these are both federally endangered species here. And the landowner didn't even live in the state and had absolutely no idea what was going on. And when they were made aware of this, they worked out a deal with Heritage Trust Program to set that aside as an annex to the adjacent heritage preserve. And so if we didn't have this specimen here, which is not duplicated at any of the other herbaria, it would not have been sort of a proof that these specimens were, these species were present on that property. Furman University, which is largely inactive at this point in time, has a lot of historical specimens, many of which were collected from what is now underneath the waters of Lake Jocassee. And they also have some other very rare specimens of endangered species that are only known from one location in South Carolina, like this this orchid here, Platanthera integralabia, and the purple mountain pitcher plant, and shortia, what do they call it? Oconee bells would be the common name. And right about now, if you want to go see those things, it's about time to visit the upstate. But anyway, so no other herbarium in the world has representations of these species. This orchid in particular is known from a single population, and this is like the only representative herbarium specimen of it occurring in South Carolina. Appalachian State University in Boone, they had records of shortia, Oconee bells, that is otherwise apparently lost. Again, this might be one that's under the waters of Lake Jocassee. And then there was this really interesting circumstance where Catawba College was not part of the original CERNEC proposal. It was a small college in North Carolina that had a herbarium, and I was friends with a person who's part of the North Carolina Natural Heritage Program, and he had mentioned visiting this herbarium in person so that they could search through the collections for any previously undocumented populations of rare plant species. And so I reached out to him and Dr. Baranski, who's the curator there at Catawba College, and said, hey, you know, we can help you get this stuff online if you've got records in some sort of digital format already. We'll be happy to, you know, facilitate the process. And sure enough, he had just a simple spreadsheet, and it was, the data were parsed out in a relatively nice way. And we were able to go from 0 to 5,214 specimens in less than 30 seconds. And so now those data are accessible to anybody in the world. And I think it just, it speaks to the importance of these smaller collections. So I'm going to switch gears a little bit and talk about some of the new avenues for research that we might get out of this. Of course, the traditional taxonomic studies are still going to be involved with digital herbarium data, but they might use tools like ImageJ or other morphometrics analysis software to tease out differences between species that are close lookalikes. And that's one reason that we include the scale bar in our digital specimen images, is so that they can be calibrated and you can actually get relatively accurate measurements of the plant parts and those sorts of things that you're looking at. Again, I had mentioned that the image quality that we have is the equivalent to 40 times magnification. And in my own personal experience, I've been able to use that to measure plant hairs that were on the order of 2 millimeters in length. And that actually can be sort of a way of differentiating one species from another. On a broader scale, we could look at herbarium specimen records across a geographic region and or a physiographic province, and try to characterize different communities that are present within that area. So we can use search methods that are available in the portal that allow us to draw a polygon across an area, and any of the georeferenced specimens that are within those bounds will pop up as a dot. And on the left-hand side there, where you see the bigger numbers like 300 or 100, if you click on any one of those, it's sort of like a hub and spoke. It'll expand and it'll show all the individual dots that are associated with that collection site. So this definitely makes it useful for biogeographic studies, looking at range distributions of species. We have seen some studies where they've looked at invasive species distributions. Obviously, rare species and conservation models are enhanced by having these data available. And what I did for my dissertation was looking at potential species migrations under forecast climate change. So that involves coupling these sorts of data with forecast climate models. Okay, and then there's other studies that look at phenology and changing in blooming times and whether or not that changes across an altitudinal gradient or latitudinal gradient. One such example of a study that looked at invasive species was delivered as a talk at our botany meeting in 2019. This paper is now in prep for publication. And what they did here was they essentially identified several of the worst offenders that were considered invasive plant species in Tennessee, and then characterized what sorts of habitat conditions they were associated with, and used some of the data layers available in the World Climb data set to run these distribution models in Maxent. And the result was enlightening in that some of the species were showing up as potential invaders to areas that had previously not been seen. So the idea is to kind of keep a watch in those newly identified counties that might be have conducive conditions for growing these invasive species. Another easy way to do these sorts of things, looking at phenology, is to just do a search for a particular species and sort your records by state. And so I just picked just for kicks here. I picked white oak, which is Quercus alba, has a fairly wide distribution across most of North America, or at least Eastern North America. And you can just scroll through the thumbnails in the results that are returned in the list and pick out the ones that have the little tassels where they've gone to flower. And if you click on any one of those, you'll see the label information says when it was collected. So in states like Georgia, the majority of these specimens that have the tassels on them are collected in April. But when you move further north up to Pennsylvania, it shifts to May. So we can already see that the time these plants are going to be shedding their pollen has shifted almost 30 days just because of the latitudinal difference. Some other studies have looked at a much more historical record incorporating observations by Henry David Thoreau and Alfred Hosmer. Hosmer had a farm where Thoreau was a frequent visitor. And they made very detailed accounts of when they observed particular species blooming, and that's represented by the blue dots. Those would be the earliest blooming times that Thoreau or Hosmer recorded. And then the black bars that you see for each of these species on the left-hand side are when the herbarium specimens were collected. So they're comparing collection dates of herbarium specimens to the observation dates that were recorded in the 19th century. And then the red bar is kind of their adjusted flowering time interval that they've used by interpreting the length of time that herbarium specimens span relative to the first observation that Thoreau and Hosmer recorded. And this kind of gives us the idea that there's been a temporal shift in blooming time, and that may be attributable to a gradually warming climate. There's some caveats to using this sort of data, and obviously it's incomplete. And if you recall, the initial budget for the CERNEC project was cut by almost 40%. So we were sort of strapped in how much actual data development we were going to do as collections managers. And that's still sort of an ongoing process. And I think anybody who harvests a large chunk of these data needs to be careful to keep that in mind, or at least review the data that they have accessed. Invariably, there's going to be a sampling bias. If you look at the representation of different species in the herbarium collection here at USC, you will find that apparently Richland County is the most floristically diverse county in the entire state. And that's because it's our backyard. So it's very easy to collect plants from anywhere in Richland County and have them accessioned into the herbarium here. Also that sometimes botanists like to just get to places that are easily accessible. So sometimes they'll just do roadside botany and you won't really have an accurate representation of what else is out there further away from the well-traveled lanes. And then again, it's unvetted. It's presented as is. There are tools within the portal that allow researchers to contact individual collections managers when they discover a potential error, and those can be corrected in short order. So all those need to be taken into consideration when using these data. So some other cool things that I've been thinking about involve plants and allergies, and one that affects a lot of people pretty severely is poison ivy. And there was a study done probably 7 to 8 years ago in the Duke experimental forest where they basically outfitted this huge dome over trees and everything and pumped extra CO2 under it to sort of simulate what our atmospheric conditions around the globe might be, a few years from now. And what they found was that poison ivy responds very well. It grows much more vigorously. And in fact, the urushiol, which is the allergen that it produces, is produced in greater supply. So not only is it going to be bigger and better at growing, it will be more toxic. And the map that I'm showing you there shows its current range. And I'd be also interested to see if forecast climate conditions might also point to potential for it to spread outside of that current distribution. We talk about seasonal allergies. Generally, trees like maple, oak, and hickory, pines also tend to be more of an offender during the springtime. In the summertime, we get grasses and those sorts of things. And again, all of these are wind dispersed, have wind dispersed pollen. So they're very small, very light, and can get way up into the atmosphere. And then in the summer and fall, in keeping with most things that are in the sunflower family, ragweeds come into play, and they tend to be pretty difficult for a lot of folks. And often ragweed gets confused with goldenrod. And so I think that's one thing that we as herbaria can help with when looking at these sorts of things is describing the differences in those two plants and why one probably is an allergen and the other is not. So ragweeds are in the genus Ambrosia. You can see one of these that we have here in South Carolina has the epithet Trifida. So I've included this Day of the Triffids broadside. But anyway, Ambrosia has very small, light pollen grains. It produces male flowers on the spikes that stand above the rest of the plant, and the pollen typically is just designed to sort of fall down on the female flowers, which are right in that sort of little axle that you can see in the middle image here. But again, because it's so light, it can get airborne very easily and get into folks, into your respiratory system and wreak havoc. So there's about 31 species across North America, and most of them are through the lower 48. But in searching the CERNEC portal, I found that indeed there are a few collections of Ambrosia species showing up in the southern parts of Alaska now. So potential range expansion, that could be another sampling bias issue. We don't really know, but it definitely merits some additional investigation. By comparison, there are probably upwards of 100 species of Solidago, which is goldenrod. Now, the reason this one isn't really much of an offender, and I'd like to kind of dispel any myths on that, is because it is insect pollinated. So the pollen grains are almost twice as large and designed to stick to bees and that sort of thing, and don't really fly very well on their own. And it's also widespread throughout the lower 48 and into Alaska. So I don't think this one is really one of the ones we need to be worried about. And I think that's about all I've got. I'd like to open up for questions. I've got some resources listed here if anyone's interested in accessing some of the stuff that I included here.
Awesome. Well, thank you so much for that presentation, and we will open it for questions as others are thinking about their questions, I'm curious just how to leverage this and use it. Is it reasonable to make assumptions that the changes that you're seeing can be used as a measure of the environment, right? So to if I always think about the questions that funding agencies want is, can we have additional data or additional innovation into what we're using? And how would we be able to say, hey, this is the changes we're seeing, and so that matches whether we have environmental contamination or this or that, for folks like us that don't have the kind of knowledge you have.
Yeah, so I'm trying to think of some examples of how that would work. For instance, because there's a collecting date associated with all of the herbarium specimens, one of the things we as botanists like to do is document any new county occurrences and that sort of thing. So it's also relatively easy to kind of tease out a timeline for when species were discovered in different parts of the world. And the example I like to give is turning back the clock a bit and thinking about South Carolina as a colony, and how initially Charleston being the sort of the first city in the state, and how the early natural history explorers, when they came in, would have landed in Charleston and probably not made it much further into the interior. So the earliest collections of any of the species that we have in South Carolina tend to be along the coastal plain. And through time, what you see is that all those coastal counties end up having a documented occurrence of a particular species very early on. And slowly through time, you end up adding additional counties towards the interior. And that sort of pattern, if you were to graph, it's going to follow sort of a logarithmic curve, where the initial exploration has this exponential kind of fast growth where all the easily accessible areas are well documented. And then as you get further and further inland and towards the inland limit of a particular species range, you find that your addition of new county occurrences starts leveling off. So I'm thinking that using these collecting dates would be one way for sure to tell if you've got any sort of new populations showing up outside of their range, or if we're looking at range extensions and that kind of thing. And did I answer your question somewhat?
Okay. I think we had another question from Dr. Hickman.
Good morning. Thank you for the great presentation. Lots of information and data. I may have missed it. Is it possible to take image via cell phone and then tap into your library and identify a plant that I found in my farm or my backyard?
So I think what you're asking, you're saying if you have a mystery plant that you're looking at and you grab an image of it,
Correct.
Do we have sort of artificial intelligence that can help nail that down? Not through the CERNEC portal. I know that there are a few applications or apps that you can get on your phone that make a reasonable stab at that. One of the earlier ones was something called Plant Snap, but I think it's no longer supported.
Right, those are most.
You run into some problems.
Yeah, you're right. I mean, those are most. I tried many, and those are not true scientific resources per se, and use is of course definitely scientific. And I was wondering if it is not, is there intention or possibility to add that capability, since the university has.
Right. Yeah. So that is some of the things that we are exploring, and that was one of the really neat tangential things that popped up as a result of the CERNEC project, where we were, had a lot of stuff to do in a very short amount of time with very little funding. So we needed cheap student labor, and I found it heartening that a lot of our project participants or PIs were not averse to enrolling students from different academic backgrounds. So we had some students that were largely computer programming majors and that sort of thing that ended up being converted to this hybrid of biodiversity informatics type studies. And they have looked at ways to read the specimen images and scrub them for the label information, or read the barcode values off of them and rename the image file based on the barcode value that's in the image. So streamlining the workflows. And then other students were actually trying to look at that sort of artificial intelligence application where they could program some sort of code that would look at the specimen image and identify a flower, and then try to run some morphometrics on that to narrow down like which plant family it might be in. The issue that we run into is that our specimens are dead. They're dried, pressed, and flat and squished. So there tends to be a lot of sort of distortion involved in that. So we would have to have some sort of library that corresponds to the living image and how that might get distorted when it's preserved.
That is the question, because the image I would take would be a live plant, definitely.
Right.
Small, 99%. But that would be an interesting challenge if there's a way to morph that live image into a potential dead image, right? And then to compare the dead image. I'm just imagining.
Yeah, exactly. Yeah. Yeah, and so the portal does allow the option for any user, like so any person, any Joe public can create an account in this portal, and you can create your own checklists and upload your own live plant images and those sorts of things. One of the things that we're working on here at our herbarium is trying to devise a way to associate live in situ images of the specimen prior to it being collected with the specimen. And so we're looking at using QR codes that we will then glue onto the specimen sheet, and then that will allow a researcher, if they're going through the herbarium and looking at the physical specimen, they can scan the QR code, pull up the record in Cernic, view the specimen once it's preserved, and any of the associated live specimen images as well.
It might be interesting
in terms of
crowd imaging, sourcing, getting the assistance of the people to dump images to you, create more broader database for future use, perhaps. You know, as you take images and upload, then there's a live image library per se, built on your side.
Right.
So anyway, I'll keep quiet. This is pretty interesting. I appreciate it.
Cool. Thank you.
All right. Do we have any other questions? Okay. Well, thank you so much, Dr. Herrick, for the time that you've taken. I'm sure we'll have people who will probably follow up with you just to get some more information, more detail. And that concludes our session today.